I want to thank, as the other speakers, the committees that invited me, but I want also to thank them because I think that this day on nanobiology was very interesting and with a very multidisciplinary approach, okay? And I think that this is very important, and I will start my, on the concept of multidisciplinary because I'm a medicinal chemist, and medicinal chemist is an inherently multidisciplinary topic because it deals with the design, the optimization of molecules that have to become drugs, okay, and that have to present some properties that are important in order to obtain a drug that could be marketed. So normally we are interested in pharmacokinetic, that means what the drug does to the body, okay? This means how the drug interacts with the receptor, with protein, and which is the signaling pathway that occurs after the drug has interacted with the receptor. But we are also interested in the second part that it's called pharmacokinetic and what the body does to the drug, okay? This means that we have to study the absorption, the distribution, the metabolism, and the excretion of a new molecule that has to become a drug. Very interesting is to say that normally the deficiency in ADME tox characteristics or profile are the leading cause of attrition during the drug development, as this morning Ivana described very well, because it's true that, for example, I don't know if you read it very well, but normally the pharmaceutical companies started from a test from 10,000 to 20,000 molecules, okay? And then they filter after a lot of assays and tests, and they arrive to the ADME in order to describe which are the absorption, distribution, metabolism, and excretion of a molecule. Which are the physical-chemical parameters that governs the ADME profile of a molecule? They are quite simple because they are solubility, hydrogen bonding capability, coefficient partition, and that is log P or log D, and the molecular weight, okay, of a molecule. Sometimes we have some problems concerning the ADME profile, and also this morning Ivana showed you this example. Paclitaxel is one of the most effective drugs in the treatment of different cancer types, breast and ovarian cancers, but it has a low solubility in water, and that is why in the last years it is clinically used as a liposome nano formulation, liposu, or entrapped in a nanoparticle made of albumin, okay? So nanocarriers permits us to improve, for example, the permeability of a compound that has, for example, a bad lipophilic profile. It gave us the opportunity to enhance the solubility, to provide a better prevention for metabolism and so on. This is true for biotechnological drugs also, and for the targeting. So the molecule has to arrive to a specific tissue or to a specific cell line, okay? In our lab, we used different type of nanosystems in order to overcome some drawbacks of some new molecules that had to arrive to the market. And we used different types of nanomaterials like carbon nanotubes and carbon nanodots, gold nanoparticles, haloside nanotubes that are aluminosilicate derivatives, solid lipid nanoparticles, and in the last years we also prepared some protein nanoparticles. Our purpose was to use these nanomaterials essentially as drug delivery systems, okay, in different type of cancer, for example, for prostate cancer, for breast cancer, also here in the radiotherapy for breast cancer. And as you can see here in this picture that was presented in this paper in Nature Nanotechnology, you can see that there are a lot of nanomaterials that could be helpful to improve the pharmacokinetic profile of some drugs. And as you can see, there are liposomes that are the most used in clinic, and then nanocrystals, emulsion, and so on. And the disease that is most studied from the nanomedicine point of view is cancer, okay? Because we will see that one of the problems in the cancer therapy is that the drugs sometimes did not arrive to the tumor directly, and so we have to comprehend and try to enhance the permeability and the diffusion of the drug in the solid tumors. We said to exert its activity, the drug must reach the biological target, but many cancer drugs have limited distribution, as I said before, in some solid tumors, and this limitation is done because we have some physical cues that could be a barrier for the penetration of the drug, like physical barrier extracellular matrix that normally is more dense, or biological and biochemical cues like hypoxia or the pH value that is different from a tumoral cell and a normal cell. The chemoresistance is due to the tumor environmental factor, obviously, but also it's due to the structure of the drug that we are using, okay? So we can say that several strategies have the potential to improve the penetration of the drug into the tumor, and one of the strategies is to use nanomaterials, for example, as drug delivery systems. But it is also important to assess in vitro, in vivo, the quantitative assessment of a drug penetration into a solid tumor. This is not so easy to do. I will show you why. We said, okay, we can change the formulation, and so we can modify the penetration of the drug with nanomaterials, for example, or changing the structure of the molecule that you are testing. But we have to obtain also good information on the morphology of the tumors. Normally, when we perform pharmacokinetic studies, we use a process that is quite simple, because we take the tumor for the animal, animal tissue, we perform an homogenation, we add lysis buffer, we made a purification by solid phase extraction, and then we monitor the quantity of the drug that is present in the tumor, in the tissue that we are analyzing. But this approach, with this approach, it's impossible to know where the drug is localized in the solid tumor. The drug and its metabolites also. We don't have any idea about the distribution of the drug in the solid tumor when we use this classical approach of pharmacokinetic studies. So, in the last years, it's known that mass spectrometry imaging is a very powerful label-free technique that permits us to analyze the distribution in ex vivo tissues, okay? And in particular, there's a technique that it's called MALDI-TOF mass spectrometry. It means matrix-assisted laser desorption ionization. It's used a lot in preclinical and clinical studies, okay? Very rapidly, I will say to you how it works. So, MALDI-TOF mass spectrometry is based on a three-step process. The first one is to mix the analyte in which you are interested in with a matrix, okay? The matrix is able to ionize your analyte easily, and before you have a desorption and an ionization, that is the first step, then you go to the mass spectrometer here, and you have the mass analyzer and the ion detector that permits to obtain some information about the molecules and the metabolites, the drugs and the metabolites, and also information about the quantity that is present in your sample, okay? This technique was discovered by Tanaka. He won the Nobel Prize in 2002, okay? And so we thought that this technique could be very useful in order to obtain some information about the drug distribution in a solid tumor. Normally, the matrices that are used in this technique are organic compounds. They are small molecules. Normally, they are acid, okay? But this technique was useful, using these matrices, only for molecules with a high molecular weight. This means peptides, oligonucleotides, proteins, and so on. But when we have a drug that is a small molecule and its metabolites are small molecules, we need to change this type of matrix because this organic matrix could also give an excessive fragmentation, as I wrote here. And this means that you have a high background noise in the low mass spectra region, okay? Because also they will fragmentate and give some ions, okay? This fragmentation could mask the analytes ion, okay? Another problem is that these matrices, these organic matrices, could co-crystallize with the analytes, and so the spatial resolution of the image that you obtain are not so good, okay? That is why, for example, at 1988, so very early, Tanaka, that discovered also the Maldi-Toff technique, used for the first time some nanoparticles, in particular cobalt powder, in order to analyze some protein and polymers, high molecular weight molecules, okay? Then we have a development, and in particular a group of the Mario Negri Institute in Milano used for the first time titanium oxide as matrix, so nanoparticle of titanium oxide. They are not organic compounds. They will not mask the fragmentation of the drug that you are analyzing. So they obtain really good results, because if you compare these two spectra here, this one is made with one of the most used matrix, CHCA, and this is obtained in the presence of titanium oxide as matrices. You can see absolutely that this spectrum here is more clean than this one that we obtain with the organic matrix. So 4 or 5 years ago, we entered in a project, and the project title was Nanostructured Initiators for Matrix-Free Surface-Based Mass Spectrometry Imaging of Anti-Tumor Drugs. So this means that we want to find some new nanosystems that could be useful in mass spectrometry imaging to monitor drug distribution in solid tumors. So here there's a summary of the nanoparticles, of the nanomaterials that were used in order to identify some small molecules compounds. And you can see that we start from metal oxide nanoparticles, carbon nanoparticles, metals nanoparticles, compared to the organic matrices. And here you can see the molecules that are identified by this type, ionized by this type of nanomaterials. So we started with the most simple experiment, and we said, okay, we want to obtain some information about the distribution on a solid tumor. We will start with a 2D analysis of tumor slices using titanium dioxide at the beginning of our experiment. So we put the slice of the tissue, of the tumor tissue, on the MALDI plate. We spread the matrix, we load with the matrix. Then here, to acquire we acquire the mass spectrum for each point of the tissue section by rastering the laser beam in a defined geometrical coordinates. Okay, so we had a lot of spectrum, okay, to analyze. Then we put all together with bioinformatic analysis of the spectra, and we started with the study of the distribution of Paclitax cell. So here you can see our first results. You can see that Paclitax cell distribution varied between different types of tissues. If you look here, you have the liver, the spleen, and the tumor. Immediately, you can observe that in the tumor, the drug is not homogeneously distributed. Then we went further. Paclitax cell distribution varied also between different tumor types, xenograft, okay? So melanoma, breast, sarcoma, colon, ovarian, mesothelioma. You can see that it's really heterogeneous, the results that we obtained. Not only this, but even between different xenografts of the same tumor type, we obtain different distribution of the drug, okay? This was very encouraging because we said, okay, we can really monitor the distribution of the drugs, and we went further and we said, and we observed, for example, how bevacizumab, that is an antibody that was used, it's not used anymore, is able to ameliorate the distribution of paclitaxel in the tumor because maybe bevacizumab is able to modify the microenvironment in the tumor. So in clinic, paclitaxel is administered with bevacizumab, okay? And we hypothesized that this could be the way by which bevacizumab could enhance the distribution of paclitaxel in the tumor. Then we said, okay, everything is working, we boost our technique, we want to go further, okay? We want to obtain 3D images of the distribution of our drugs. The protocol is quite the same. There's only the last part that is a very hard part because we have to superpose a lot of images in order to obtain the reconstruction of a 3D image. This is the protocol. I will not enter into the detail because we have not so much time, okay? But I want only to mention the cell line that is MPM 487, that is a malignant pleural mesothelioma xenograft, and it's normally resistant to the anti-tumor drugs, and so we decided to work on this cell line. Then we obtained the slices when the tumor arrived to the weight of 1,000 milligrams. We cut the tumor using a cryomicrotome at minus 20 degrees, and we obtained a lot of slices, okay? And then we made the analysis using always titanium dioxide as a matrix. We obtained these results. Oh no. Doesn't work. Sorry. Before it was going, okay, sorry. Okay, here you can see our results. As you can observe here, the drug, the paclitaxel, is present only on the edge of the tumor. It never arrives in the center of the tumor, okay? This means that only a low part of the drug, a very the minimum concentration, a minimum concentration of the drug arrives to the solid tumors, and the drug that arrives is accumulated on the edge of the tumor. Okay. Then we said, okay, this is important, but we have also to make a quantitation of the drug that is present into the solid tumor. And as you can see here, look at number 1. Here you can see that paclitaxel is present. And then when we go to the center of the tumor, of the center part of the tumor, you can see that paclitaxel will completely disappear. Okay? We compared also the data that we obtained by simple LC mass analysis that we saw before, the typical pharmacokinetic analysis that we made normally by homogenation of the solid tumor. And we can say that, okay, we obtained quite the same quantity, but the data that we obtained with this simple approach will not say anything about the distribution, okay? So we could quantify the molecules, the drug that arrived to the tumor, but we are also able to say how, where it is. Then we said, okay, we have wonderful results, but we want also to test other nanomaterials, not only titanium dioxide, and we use some materials that we used also in our labs for drug delivery systems, like gold nanoparticles, haloside, and carbon nanotubes. And we used not only paclitaxel, but different types of drug with different mechanism of action. These drugs are anti-tumoral drugs that are used in clinic. So all the nanomaterials were firstly screened to compare their ionization efficiency, efficiency with different type of drugs. And as you can see here, the most important result was obtained with nanogold nanoparticles with imatinib. And that is why we went further and we made a paper only on imatinib and gold nanoparticle. And as you can see here, as we saw for the paclitaxel, also in this case, using MALDI-TOF spectrometry and gold nanoparticles, we are able to know where the drug is distributed. And it will never go in the necrotic and fibrotic part of the tumor. So my conclusion is that we tested different nanomaterials and that it's possible to overcome the problem of high background noise that normally is present with the typical matrices used in MALDI. Now we are testing new nanomaterials that are functionalized. We made some hybrids of different systems, nanomaterials, in order to test this type of materials with this technique. So at the end, I want only to thank Enrico Davolo and his group of the Istituto Mario Negri of Milano, Falciola and his co-workers of the University of Milano, and Pietro Franceschi from the Fondazione Edmund Mack. Thank you very much for your attention.